In today's video, we're going to finish off chapter 11, where we've been talking about memory mechanisms. We started off the chapter talking about different kinds of comparative setups, different experimental designs that we could be using, including things like mazes and delayed matching to sample and so on. And then we started talking about how we can actually use those setups to test for different abilities within different species. Of a location where things are within an area, so landmarks, elevation, whatever aspects they might be including. We also commented on the fact that our cognitive maps, even if we talk about animals potentially using them, we can't really pick apart exactly what those maps are storing. We don't know what information is contained in that map, and specifically, we don't know how the animals might be using that information. So what we might choose to focus on instead is things like a navigational code, where we can code information using, say, specific cues or specific mechanisms. So that's what we're going to start looking at on the next couple of slides. So if we start talking about coding in a natural spatial task, we can talk about beacons and landmarks, which would both be kinds of navigational codes because we might store information about items, stimuli, that can act as a beacon or a landmark. So when we're talking about beacons and landmarks, a beacon is just a spatial cue that is located right next to the goal. So if we look at the bottom here, we have a rabbit, and there's a food cache that they might have hidden or that they might have easy access to, or maybe that an experiment has made available to them. And then there's a large rock right behind that food patch. So the rabbit can look at that, and if they see that rock, if they walk towards that rock, they will find the food patch. So that rock acts as a beacon. And beacon following is a very simple navigational tactic where an organism can learn to associate that beacon with the goal object. If you wanted to test for beacon following, if you wanted to test if an animal is using a beacon to find the goal location, in this case to find where the food is, what you could do is set up an experiment where you just move the beacon. If you move the beacon from the left side of the enclosure to the right side of the enclosure, does the animal now start searching at the other side of the enclosure? Do they follow the beacon? And if they do search in that new beacon location, then you can conclude that they're probably following the beacon. Now, we can also talk about landmarks. And landmarks are also oh boy, I'm having trouble with words today. Landmarks are spatial cues that aren't directly on top of or right next to the goal location. So they're more distant and distinct from that goal location. So a landmark would be something like this bush or those smaller bushes over there, which are off to one side. So a landmark is still a location that can help you figure out where the goal is, but it's not as simple as just following a beacon. So with a beacon, you just head to the beacon and you find the goal. With a landmark, you now have to learn where that landmark is in relation to your goal, and you have to know how far away it is and what direction and so on and so forth. So if we were looking top down onto an enclosure, if we have a beacon here, then the animal could go to that beacon and find the goal. Whereas if I swap over my color, maybe we have a landmark and a landmark here. So they would have to learn that they can't just go to the landmark. They have to find the location that's equally distant between the two landmarks. So if they wanted to find that goal location, they'd have to know that it's, up and to the right from this landmark, and it is down and to the left from that landmark. So landmarks are a little bit more complicated to use than just a beacon. But you can test whether they're using a landmark in the same way that you test if they're using a beacon. If you move the landmark and then see where they start searching, if they adjust their search location when the landmark is relocated, then they're probably using that landmark to find the goal. And this is just an example of what it might look like in an experimental setup rather than our cartoon. This is from Eric Legg's lab. And so we have a pigeon here, and the pigeon is trying to find some food that's in this central location, and this green pillar can serve as a landmark. So looking at that green pillar, they can figure out that they have to be this far away from it in that direction to find a food location. Whereas the beacon over here, I believe in this setup, the goal is to find this opening in the enclosure, and this post-it note would serve as an indicator of where that hole is. So if you go to the post-it note, you have found the goal location. So that post-it note would serve as a beacon, whereas this pillar here would serve as a landmark. Our next kind of navigational coding strategy that we can use is geometry. And so with geometry, you're using the shape of the room or the enclosure that you're in to locate a goal. So in this instance, we have two different types of corners in this rectangular room. If we're using geometry, we're just going to be using the angle of the corners and the lengths of the wall, basically the shape of that room, in order to determine where to look. So if we focus just on this one corner here in the top left, we have on our left-hand side, if you're facing into that corner, the left side is a short wall. And if you look to your right, then you have a long wall. So short left and long right would put you in this corner. But those same determinants, those same rules would also work if you were facing into this corner. This green corner here also has short left and long right if you're facing into it. And conversely, if we focus on the red corners are the opposite, where it is long left, short right. But these two corners here, circled in red, are both geometrically equivalent. So if we were expecting this pigeon in this rectangular room to be relying on geometric cues, we would expect that they would split their searching, they would split their sort of goal location assumptions between the equivalent corners. So if they're used to finding food in this kind of a corner, we should see that they'd also search in this kind of a corner as well. And if they're relying on geometry, these two corners should be searched quite a bit less because they're not the same as locations that they've previously found food. So let's look at an actual experiment that's set up in this way to look at geometric cues. So this is an experiment looking at geometry and featural information in a spatial orientation task. So the way that this experiment is set up is that they were looking at two different species of chickadees. We have black-capped chickadees and mountain chickadees. They're both fairly similar. They actually overlap in habitat a little bit, but these guys, as you might expect from the name, mountain chickadees live in the mountains, and black-capped chickadees tend not to live too close to the mountains. So we just wanted to see if they have similar use of geometry and featural information, so we looked at both. And just to be extra fancy, they also looked at hand-reared black-capped chickadees. So wild-caught birds would be birds that we went out into the field and caught out in the wilds where they had been exposed to nature and natural environments, whereas hand-reared birds would have been raised in the lab without exposure to those natural environments. And the way that we're going to read these diagrams is that, okay, so we have a rectangle. That means that there was a rectangular room in which they were trained. The star indicates the correct corner. So this would have been the corner that was baited. I believe in this experiment we used mealworms, so a cup with mealworms in order to get the chickadees to search in that corner. And they just measure, that's what these numbers are. These numbers are a measure of how many times the chickadees searched in each of these corners. So you would have a cup full of mealworms hidden under some sawdust at the bottom, so they would have to dig so that you could measure digging behavior to indicate a choice made. And we see that about 50% of the time, these birds were searching in the correct corner. And any errors that they made, most of those errors were to the geometrically equivalent corner. So we see a couple of errors, 5 and 7, in these two corners, and I believe these are just percentages, so a total of 12% of the errors were to a non-geometrically correct corner, but the vast majority of searching behavior was done in the corners that were either correct or geometrically equivalent to correct. And we see this pattern across whether the birds were hand-reared and or wild-caught for black-capped chickadees or mountain chickadees. We are seeing the highest responding in the correct or geometrically correct corners. So that tells us that these black-capped chickadees, when they're trained with geometry, are fairly good at finding the geometrically correct corner. Okay, what happens if we introduce a feature? In this instance here, the star is still showing us the correct corner, but now we've introduced a wall. And so this would be a colored wall. And so because we can now use a featural cue, a feature being a colored wall, as well as our geometry, now we can distinguish between the two geometrically equivalent corners. Because, yes, this one was short right and long left, but now it's short right and purple wall, whereas this one is still short right and long left, but it doesn't have a purple wall. So that would tell us that this is not the correct corner. So now we see about 97% of searching was done in the correct corner because this feature has allowed us to distinguish between the two geometrically equivalent corners. So the chickadees can use geometry and featural cues to find the correct corner. Now we can move on from that and extend this sort of interaction of geometry and feature cues to see what they use. We can set up a setup like this, where we always have purple on the left and white on the right. So this corner, purple left, right, white right, and so on and so forth. So the feature in this case isn't helpful, but the geometry is helpful. And we come back to that issue where they go to the geometrically correct corners again. So this is a probe trial where there isn't actually food present, but it's based on what they had been trained where this would have been the correct corner. So we're just seeing that the geometrically correct corners are searched in more often than those that are incorrect. So if we had been, or if the chickadees had been using only features, if they had only relied on the wall color, then they should have split their searching equally between all corners, but they didn't, so they're using geometry. And then we get into the cool stuff. So what happens if we set up a conflict? So this is what they had been trained on, where they had the purple wall here, and this was the correct corner, but then we adjust it, and they're tested in a room where the purple wall is actually a short wall, and it's in a different location. So if the birds were using features, if they preferred to use feature over geometry, then this top right corner would be correct. And we actually do see more searching in that corner. So they are still using feature cues in this situation, even though we didn't see that here. And then if they were using geometry before features in this conflict setup, they would have searched in either of these two corners, which is what we saw over here. So what we've found is that when we put these two things into conflict, they do go for the featurally correct corner, but most of their errors are made to the geometrically correct corners. So the chickadees are using both geometry and featural information here, which is really, really, really cool. And so this was for our black-capped chickadees. So I can sort of scoot these graphs all off to one side here like this. So we still have our control group where we were trained, where they have the feature and the geometry working together, and they choose the correct corner very well when they can use both features. When geometry alone is reliable, they can split their searching fairly evenly between the geometrically correct corners. And then when geometry and featural cues are put into conflict, they rely on that feature cue the most, but they split their errors between the geometrically correct corners. But how do our mountain chickadees compare? So for our control group, they look pretty much the same. They're mostly going to the correct corner. It's fine. Where it starts to get cool is when we look at our geometry condition. So when we have each corner is technically featurally correct, we're actually seeing more responding to all corners. So with our black-capped chickadees, it was mostly responding just to the geometrically correct corners because feature wasn't helpful. But our mountain chickadees are almost doing a semi-even split between all of these corners. So they are still slightly higher in their searches in the geometrically correct corners, but that's a lot of searching in the geometrically incorrect corners. So this tells us that they're probably relying a lot more on feature. And we do see a very similar observance here for our conflict stage, where they are relying on that feature corner. When feature and geometry are put into conflict, they're relying on feature rather than geometry, but their errors are majorly in the geometrically correct corners. So similar when things are put into conflict, but they seem to have a higher reliance on feature. And this was research that was done with the lab that I did my research with, so we've had lots and lots of conversations about what might be going on here. And our assumption is that mountain chickadees tend to nest out in the mountains, out in the wild. And in a natural environment, there's a lot more changing in terms of, say, geometry. They don't see a lot of squares. They don't see a lot of stationary objects. Our black-capped chickadees are found in cities. They're kind of used to boxes and things that are fairly stationary. But our mountain chickadees out in the mountains might be more used to relying on things like individual features. Maybe they navigate by following a landmark or beacon. Maybe they focus on, that tree is helpful in locating an object, but they don't have to worry about two different walls and the angle at which they intersect. That's kind of our maybe a little bit anthropomorphized interpretation of what might be going on. But the results are pretty cool because we get to see that these two very, very closely related species have different preference in the types of cues that they use in these experiments. Okay, so we talked about how we might encode that information and represent that information, cognitive maps versus navigation codes. But next we want to talk about whether an animal remembers where it has been or where it has yet to go. So specifically looking at retrospective and prospective coding. So retrospective is a little bit easier conceptually for me to grasp, and this is the idea that you're remembering where you have been. So retrospective means you're thinking about past events, you're thinking about things that have gone past. Now, retro meaning things that are older, so you're remembering things that happened previously. Whereas prospective is looking into the future. It's imagining future events. So if you're using prospective memory, you're thinking about where you need to go. There's been lots and lots of study on whether animals use prospective or retrospective memory or both, and how do they decide which one to use, and at what rate, and when do they switch, and do they switch, and all sorts of questions. But we're going to focus on one example, and we're going to think about our rats in a radial arm maze. So at the very beginning of this talk about coding, we had said, okay, so this rat has a radial arm maze, and there's a whole bunch of arms that they can visit, and they don't want to visit an arm that they've already visited. So it's unlikely that they keep a list of all of the arms that they have encountered, but maybe they are doing something kind of like that, or maybe they're using their mental map to keep track of things. But are they focusing on remembering which places that they have already visited, which arms have I already been down and therefore shouldn't go down again? Or are they framing it as which arms have I yet to visit? Which ones should I go to next? Which arms am I going to go to in the future? So this diagram does a great example of illustrating that point. So we're going to focus on this retrospective coding dotted line that goes up like this here. So if you're focusing on retrospective coding, you're going to remember each of the places that you have already visited. And so for our graph here, we have our memory load, which is how much you are holding in your memory at that moment. So maybe it's your working memory. As you are working to navigate, you have to keep in mind where you've already been so you don't go back. And then across the bottom, we have number of places that have been visited. So if we just follow that dotted line there, as you visit more places, your memory load should scale up accordingly. So if you haven't been anywhere, you don't have to remember anywhere. If you've been to one place, then you have to remember one place, and so on and so forth. So the more places that you visited, the more that you have to remember. If we then look at a prospective coding interpretation of this, you remember where you need to go. So that's our solid line here. And same thing, at the very beginning, if you haven't been anywhere, then there are 6 distinct places that you could go in the future. Once you visited one of them, now there are 5 places that you could go in the future, and so on and so forth. And then by the time you get to the end, once you've visited all 6 places, you don't have to remember any places because you've already been to all of them. Now, the best strategy, the proposed best strategy, would be to switch from retrospective coding to prospective coding at that halfway point. This would be the best strategy to reduce memory load, because at the beginning, prospective coding involves a lot of memory load. At the top here, there's lots and lots of memory load for the prospective coding. And when we get to the end of retrospective coding, there's a lot to remember there too. So this strategy is saying, okay, we'll just focus on these lower levels. So at the beginning, start with retrospective coding. When you start, you don't have to remember anything because you haven't been anywhere yet. When you visited one place, you remember one thing, 2 places, 2 things, 3 places, 3 things, and then you switch. Then you acknowledge, okay, I've been to these 3 places, now I have 3 left. So if there's 3 left, and you go to the 4th place, now there's only 2 left. If you go to the 5th place, now there's only 1 left. And the 6th place, there's nothing left to remember. So by having a flexible strategy like this, your memory load never goes beyond this arbitrary value of 3. You never have to remember more than 3 places because you switch strategies before you'd have to remember larger numbers. So is that what actually happens? We're going to be looking at the data from a study by Kestner and Despain done in the 80s, where they wanted to test both rats and humans on which strategy they're using. Are they using prospective or retrospective coding, or both, like we might have predicted? So from our understanding of how these memory load things work, we would say that as memory load increases, the number of errors increase. And as memory load decreases, the number of errors should decrease. So we can use our prediction of sort of how many arms have they been to, how many arms do they have left, and we can then make a sort of correlation between the number of errors and our memory load. So if we saw that the rats were making lots and lots of errors at the very beginning, I guess we'd have to wait until they were 1 or 2 locations in. If they were making lots of errors at the very beginning, we would conclude logically that they're using prospective coding because that's the highest memory load for early on. And if we were seeing lots of errors at the very end, we would say that they were probably using retrospective coding because that has the highest memory load at the end. And if we don't see a lot of errors at the beginning or the end, if all of the errors are occurring in the middle, then it would match this strategy where you switch between retrospective and prospective memory in between. Okay, so to the actual experimental setup. So we have a 16-arm radial maze. I'm going to talk about the rats in more detail. For the humans, just know that they did an equivalent task, but on a computer as opposed to having to run in an actual maze. But so the rats had a 16-arm radial maze, and the researchers allowed the rats to enter a different number of arms on different trials. So some trials they'd enter 1, sometimes 2, sometimes 10, sometimes 12. So multiple rats with multiple trials, and each time they'd be allowed to enter a different number of arms so that we can see how their performance varied, how their errors varied after they had experience with only a couple of arms or a lot of arms from our graph. Once they had been allowed to enter the set number of arms for that particular trial, the rat would be removed and held somewhere else for about 15 minutes or so, just to give them a break to separate them from that situation, and then they'd be reintroduced to the maze. When they were brought into the maze after that break, they would be permitted to enter one of two arms. So the experimenters would block off all but two arms so that they had to make a binary choice. So one of the arms was an arm that they had previously visited and therefore wouldn't have any food in it, and the other arm would be a new arm that they had not yet visited. So the correct response would be to enter the arm that they had not entered before. If they entered one that they had already visited, that would be counted as an error. And of course, we're interested in the number of errors that they're making. So our graphs here show the mean number of errors on this axis here, and across the bottom is the number of places that they had visited before the test. So this would be the number of arms that they had been allowed to enter before they were removed and then later brought back. And the interesting thing that you might notice here is that both of these graphs are showing a peak, not at the beginning or the end, but they're showing a peak in the middle. The rat data, for whatever reason, doesn't go up to a higher number of arms. So if it was a 16-arm radial maze, they could have done up to 14 arms probably without any problem. But for whatever reason, they just didn't do that high of a number of tests beforehand. So this is at 8, which is halfway through the 16-arm maze. So we see the greatest number of errors at that midpoint, where they're carrying the highest memory load if they use that switching strategy. So this would be evidence that the rats and humans are switching from retrospective coding to prospective coding after 8-arm entries, which means that at that 8-arm entry point, that's where they have the highest memory load, and that's where they're having the most errors. All right, so that was acquisition and encoding. Next, we're going to move on to retention and storage. Now, one of the biggest issues that you have to deal with when testing for retention is the issue of rehearsal. Rehearsal is kind of just what it sounds like. If you're given a grocery list and you have to walk 2 minutes to the store and you don't want to write that list down, maybe you're going to repeat that list over and over in your head so that you remember it. So as you walk, you can rehearse that list and keep it fresh in your memory so you don't forget. The issue here is that if you're trying to test retention and someone is rehearsing, then you're more getting a picture of their working memory ability and not their long-term memory ability. You're not getting a sense of their retention, you're just getting a sense of their ability to rehearse and hold information active in their working memory. So this works for humans and non-human animals, where if you want to get the best picture of what they are able to retain without rehearsal, you have to somehow distract them from using that rehearsal process. Some of the neatest studies that have looked at rehearsal and not using rehearsal come from studies that are based on directed forgetting. So the idea here is that you can have two different kinds of cues, F cues and R cues. R cues are cues to remember, F cues are cues to forget. And so remember cues are signals that let you know that you should remember this information because it will be tested later. And if an individual's memory ability is tied to their rehearsal, being told to remember some information should lead to rehearsal, which should lead to better performance. If someone is given a forget cue, an F cue, then we're being told that you're not going to need this information later, you don't have to hold on to it, you don't have to rehearse it. If we then test their remembering of that information, once they have not been rehearsing it, we should see a reduction in performance because they haven't been rehearsing that information, they haven't been keeping it active. So let's look at an example to try and solidify what I'm meaning with that information. So this is a delayed matching to sample task done in pigeons. And we're going to be looking at 3 basic types of trials. We have a remember trial, a forget trial, and a free reward trial. This free reward trial is here because sometimes memory is better if you get food afterwards. Because of that extra association where this is something that will lead to food, we tend to see a spike in memory with these kinds of trials. So it's just included here as sort of an extra control. So for our remember trials, we have our intertrial interval, basically waiting for the trial to start. We then see a sample. So if you remember our delayed matching to sample, there's 3 keys here. In the middle, we are given the sample that we may or may not have to match with later on. After we've seen that sample, then we would get a cue. For the pigeons, it was a tone, either high-pitched or low-pitched, or something that varied. So a free reward trial tone here is somewhere sort of in the middle, and it kind of warbles between 2 sounds. So this one is very distinctive from the remember cue and the forget cue. So in our remember cue trial, we saw our sample, we got the sound that says remember that sample, there's a delay, and then we're given a test. Can you match your sample to these 2 different options? And if you click the correct thing, the color or the key that matches your sample, you get a reward. For our forget trials, we have our sample, we have our low pitch, there's a delay, and then we have our intertrial interval again. There is no comparison trial here. So we're teaching the pigeons that when you hear this forget cue, you aren't going to be asked about the sample that you just saw. There is no comparison, so you can forget that information. Whereas up here, when we have a remember cue, you're going to need that information later, so you had better remember it. For a free reward trial, sample, the free reward sound, a delay, and then you just get a reward no matter what. So there's still no comparison trial here, but you get food anyways. And if we want to see how effective these cues are in controlling memory, we can use what's called a probe trial. And this is when you introduce, every once in a while, not every single time it happens, but after you've trained up these remember, forget, and free reward trials, you introduce a couple of tests. Tests or probe trials. And so here, our forget probe trial, which should be similar to this guy, has our sample, has the forget sound, there's the delay, and then instead of launching straight into that intertrial interval, then the birds are offered a comparison. And if they get the correct answer, they receive a reward. But they were told that they don't need to remember that information. So the idea here is that they should perform less good on this comparison because they haven't been rehearsing or remembering that information, that sample, during this period. And we also have a free reward probe trial to see how they perform after a delay if they are used to just getting food without having to compare. But now we're asking them to compare in order to get that food. So we are seeing what they've learned about these cues. Was the forget cue actually something that told them to forget that information, or did they remember it anyways? And we see kind of what I set us up to expect from our description. Our R cues, the remember cues, they tend to have pretty high accuracy. These codes are just for 2 different birds. So the birds tended to perform at, so this guy had about 80% accuracy, this one had about 90% accuracy. That's pretty good. For the F cues, the forgetting cues, we see that they are less accurate on those forgetting cue trials for the probes specifically. So they didn't perform as well as the R cues, and we see that across both birds. No matter how accurate they were to begin with, they still showed a drop in accuracy. And really interesting, we see that with our free reward trials, our free reward probe trials, we saw fairly high performance, fairly accurate performance, and that's because the anticipation of getting a reward helped them keep that experience in their memory. So that introduction of food also led to better performance. And because we're focusing on retention here, I want to point out that we can't say that our forget cue was, say, interfering with acquisition or encoding, because the birds were exposed to the same cues, or to the same stimuli, the same samples, whether they were in a remember trial or a forget trial or a reward trial, the sample presentation was always the same. So the acquisition and encoding stage of things was always the same, but what was disrupted here was it's interrupting rehearsal. It's interrupting their ability to retain that information long term. So the sample disappeared before they were told whether they had to remember it or not. So we weren't really affecting how much attention they were paying to the sample or anything like that. This didn't interrupt their acquisition, but this did interrupt encoding. And so it shows us that rehearsal can play a major role in what we do and do not remember. All right, so we've covered acquisition, we've touched briefly on retention. Now we're going to move into retrieval. So this is recovering information so that it can be used to guide our behavior after the fact. Now, this retrieval process is of special interest because lots of memory failure is actually from deficits in our ability to recover information. So retrieval failure is one of the biggest sources of memory failure. It's not usually something to do with failure to accurately or to properly store information. It's usually that we fail to retrieve it in that moment. Now, most retrieval research focuses on reference memory research. So it's looking at sort of that long-term storage. Things like rehearsal, which we just went over, are looked at through working memory. So it's not something that's been put into long-term storage yet. It's something that's still in that earlier working memory or short-term memory stage. Now, as I said, retrieval is important because a lot of our memory deficits, a lot of the times when we forget things, it's not that that information is completely lost, it's that we haven't been able to go in and recover that information accurately. Now, we can aid in that retrieval process by introducing some of the cues that were present during the acquisition of that information. So the more cues that we're exposed to, the better our retrieval. So we can consider these retrieval cues as reminders, little hints that help us recover information that we stored away. So the more similarities between the environment that you're trying to remember something in and the environment in which you learned that information to begin with, the better your odds of successful retrieval are. This is why you might have heard that you should study in the place that you're going to take a test, because then you have all of those spatial cues the same. So it can help with recovery, with retrieval during the test. This experiment was done to test the use of retrieval cues in infants. And so our researchers wanted to see if the stimuli present during acquisition could be used as retrieval cues when they were testing later on. So specifically, they were asking, can a cloth liner be used as a retrieval cue for instrumental response? So the way that they set this experiment up is that they were using 6-month-old infants at their home. They had a crib, and in that crib, they had a seat that had them sort of sitting reclined, looking upward. And above them, they would have a mobile, basically one of those spinning things that has, say, toys or images or bright colors or something that they can look at. And to an infant, a spinning mobile would be reinforcing. So in this setup, the children or the infants would have I think it was a ribbon that was tied to their ankle so that when they kicked, the mobile would move. So kicking would be reinforced by moving that mobile. And while they were learning, they had a cloth liner in their crib surrounding them. So this crib would sort of, just imagine a baby sitting in an open-topped box, and the walls would be covered by that liner, and the liner would have a distinctive pattern on it. And so the researchers here are just going to modify what liner they were exposed to while they were training. So once they were learning that the instrumental response of leg kicking is reinforced by the mobile moving, and then what the babies did if they were in the same kind of liner or a different liner later on. The idea being that if this cloth liner was serving as a retrieval cue, then we would see more of our instrumental behavior, more of our leg kicking, when we tested them later on, when they had the same environmental stimuli, when they had the same cloth liner, than if they had a different liner. So we can actually look at the results here, and this is an obnoxious line graph. But the way that it's set up is that our first bar here is looking at the same liner in testing as there was during training. And so we have our retention score, which is just how much kicking are they showing. So a higher score here means that they are kicking more, which means that they are, I guess, remembering that instrumental response as being reinforced with mobile movement. So the children, the infants who were trained in an environment with one kind of cloth liner and were tested in the same environment with the same kind of cloth liner, if it had the same pattern on it, they showed a lot more kicking. We can then look at our different group here. So these infants were trained with one cloth liner and tested using a different liner, and specifically a different liner that they had never experienced before. So a different and novel liner. And they showed much less kicking here. So we would say that there was no context cue present here to help them remember that kicking would move the mobile, so they kicked less. Ooh, I'm really struggling with words today, I apologize. Our next weird bar pattern here is for the different but familiar group. So this group was trained with one cloth liner and then tested using a different liner, but they had previous experience with that liner, so it wasn't completely new to them. They had just never had any training with it. And we still see much reduced kicking, even though that liner was familiar, because it wasn't the same one that they were trained with. And the last group here is the non-familiar, so they had no cloth liner, so their crib had no lining to it, which meant that they could look around their familiar toy room, their playroom, so it was still familiar, but there was no liner present. And so we're seeing a very distinct difference between the group that had the same context cues and the ones that had different context cues. And the presence of that same liner led to a higher occurrence of that originally trained behavior, that instrumental kicking response. So this supports the idea that the liner serves as a retrieval cue and helps these infants retrieve what they learned during training, because testing, they have a similar condition, they have similar cues present. Now, that example talks about a situation where the retrieval cues are present only at the time of testing. So we had the environment match at testing in the same way that it did during training. But we can also look at retrieval cues through the lens of memory priming. So this would be a pre-exposure to those retrieval cues before the time of testing. So in this situation, we would have the retrieval cues presented at the end of a retention interval, but we don't actually test their memory until the next day. So if we had we can use the baby's example. They were trained that kicking in that environment would turn the mobile, and then we were going to wait a week. We were going to come back a week later. On day 6, if we were using a memory priming setup, we would have that retrieval cue presented at that 6th day, and then we would test on the 7th. So there's the 6th day is the day before the end of that interval, and then we would test immediately the next day. And the idea behind priming is that you are reactivating the memory after that retention interval. So we want to see if doing this reactivation process by priming the day before can actually increase how long something is remembered. So this data here is actually from an extension of that babies kicking to turn a mobile experiment that we talked about earlier. But in this situation, instead of focusing on the cloth liners that were in their cribs, these researchers were focusing on a smell, a distinctive odor that was present when the child was learning that kicking would turn the mobile. And then also we could use that odor or similar odors when we were priming and then testing. So in this situation, they used either cherry or coconut. So we're just going to call it generic odor. They used multiple odors to make sure that it wasn't just a response to one specific smell, but we're just going to talk about the same odor or a different odor. So during training, the babies would have been trained in that same setup in a crib, looking up at a mobile, ribbon on their ankle so that when they kicked, the mobile would turn. And so they were all trained with a particular odor during that training session. And then we waited for 6 days. On the 6th day, most of the groups were given priming. So the babies would be put in their crib, the mobile would be moved by the experimenter, and it would be either in the presence of the same odor, a different odor, or no odor. We also had a control group that received no priming. So they didn't come in on that 6th day. They just came in on the 7th day to do the testing side of things. So on the testing day, all of the babies were tested in the presence of the training odor. So they were still having the same retrieval cue that was present when they were trained, but because we're testing 7 days later, that's usually outside of the range that these infants could remember. And if we look at our no priming group, we see what we would have expected, which is very little kicking, even in the presence of the scent that they were trained in. So this tells us that 7 days is longer than a baby would normally be able to remember an operant or an instrumental conditioning experiment. So where they learned that kicking turns the mobile, 7 days is too long for them to remember that. But we do see that when the babies were primed on the 6th day in the presence of the same odor and are then tested on the 7th day again in the presence of that same odor, we do see a significant difference. So you can see there's a little asterisk here. This is the only bar that has an asterisk. So this is the only bar that showed a significant amount of kicking as compared to our no priming situation. These ones, even though the bars are fairly high, statistically, they aren't any statistically different from the no priming scenario. I always try and explain that, especially on graphs that annoyingly don't include error bars. What that usually means is that these guys probably have fairly large error bars that might look something like this, telling us that this data kind of overlaps with this group. Whereas these guys might have a much smaller set of error bars, which is why it's significantly different. But that is a whole aside that I suppose we don't really need. What we do care about here is that when there was no priming, 7 days is too long for the babies to remember that instrumental conditioning, but if we introduce that priming session during which they were exposed to the same odor, and then we test them the next day, again using that same odor, they show a significant amount of kicking, and it helps them remember what they were trained on. So yes, we can extend, how did we word it here? We can extend or increase how long something is remembered even after a long retention interval using this memory priming paradigm. All right, well, now we're going to turn away from studies on memory to focus on the studies of forgetting, or as we're going to start calling it, hopefully, memory failure. So usually you'll see forgetting as the failure to remember previously acquired information. But from the experiments that we just looked at, we've realized that not remembering information that you previously acquired might not be happening because you have permanently forgotten that information. It might just be that you didn't have the correct cues present to help you remember those pieces of information. So the term memory failure might be more accurate because it doesn't imply that it's a permanent forgetting. We're just saying that we didn't have the right situation at the moment to recall that information. So it isn't forever lost, it was just not retrievable at this moment. Now, forgetting is one of those weird fields where some people study forgetting extensively because it relates to memory. Some people think that forgetting on its own, in its own right, is an important thing to study. So you'll see different researchers take different approaches when they come at forgetting. But we can all, I hope, acknowledge that forgetting can be useful because it might allow an organism to adapt and be flexible if situations change and they have to learn something new. So if an organism is in an environment where they've had to learn to do something in a very particular way, and then something happens, maybe the climate gets hotter or drier or wetter or whatever, and as the environment changes, they need to learn to do whatever they're doing to get food or shelter or whatever in a different manner. So it might be useful to forget the old way of doing things to focus on the new way of doing things so that you don't have a competing response where you're not sure if you should do the old or the new way. If you forget the old way, then you just do the new way, no questions asked. So there's lots of talk about forgetting being biologically relevant and potentially useful in terms of survival. But again, that's its own can of worms. So if we want to talk about why animals might forget, we can talk about different kinds of interference and then move on to amnesia. But we can talk about first our interference types. We can have proactive and retroactive interference. And this is one of those where I always forget which one is which. For whatever reason, it doesn't stick for my mind. But the way that I work it out is, again, visually, because that's the kind of person that I am. So maybe we have salient stimuli. You have to learn about A and B. If we're looking at proactive interference, we're saying that for proactive, the earlier learned item interferes with things that are learned in the future. So the learning of A would interfere with your ability to learn B later on. If we were looking at retroactive, we're saying that learning B interferes with something that you learned previously. B interferes with learning A previously. So pro meaning it's the forward moving, so A interfering with B. Retro meaning we're moving backwards, where B is interfering with A. That's just my way of remembering it. You might find your own technique to do that. But either of those situations can happen. If one or other of the stimuli is extra salient, it can interfere with the acquisition of something that comes either before or after it, and it can make it harder to remember. We can also discuss amnesia. Of course, we're going to discuss amnesia as it relates to forgetting. There are two types of amnesia, at least two types. There's retrograde and anterograde amnesia. We're only going to be focusing on retrograde because that fits with our discussion of the acquisition, storage, and retrieval of information. So for retrograde amnesia, we have greatest amnesia for events that occurred at the time of injury. So we're saying that you can't remember things that happened in the past, and specifically you have the most trouble with remembering things that happened around the event that caused the amnesia. And this can be caused by severe head injury, or we can start looking at things like animal models where we can introduce retrograde amnesia. And we're going to talk about a really, really cool study on the next slide using hypothermia to induce amnesia. And most of our animal studies of amnesia are focused on understanding consolidation. And consolidation is the process of moving information from our short-term memory into long-term memory. So how do we move our information from our active memory, stuff that we're actively using or recently using, into our long-term storage, something that's less active and for longer storage? So here we're going to look at an experiment that's going to help us pick apart whether something's happening due to consolidation problems or retrieval failure. So consolidation issues would be that we're having trouble moving information into long-term storage, whereas retrieval failure would be that we're struggling to pull things back out of storage. So kind of on either side of our list of 3 events that that are involved in memory, our acquisition, storage, and then retrieval. So consolidation would be more on the acquisition side of things, and retrieval is on the retrieval side of things. But we're going to be looking at hypothermia in rats, and specifically hypothermia that's inducing amnesia in rats, which is, as I said, so cool. So in this situation, we're using a shuttle box and we're doing fear conditioning with rats. So the shuttle box has one black side, one white side, and the rats, when they move from the white side to the black side, would be closed onto that side, the door would shut, and they would be exposed to shock. So that would introduce a fear conditioning. In this situation, it was only a single trial, so it wasn't repeated exposure. It was a one trial fear conditioning, which would mean that the rats learned that the black side is associated with shock and should therefore avoid the black side. One group was left at that stage. That's our control group. They didn't go through extinction. They were just exposed to the shock and left as is. The rest of the groups were given extinction to that black side, meaning that they were put on the black side of the shuttle box and they were left in that side without receiving a shock for a long period of time. So I believe it was 12 minutes, so that they would learn that, oh, this black side isn't actually scary. So they were extinguishing their fear. Then those groups were treated to retroactive hypothermia, meaning that we were trying to induce amnesia for the things that happened just prior to the delivery of hypothermia. And so this is done in a very controlled way where the rats were immersed in cold water to reduce their body temperature. The idea here is that that dramatic change in temperature would disrupt the process of consolidation. And so these 3 different groups, one group, the hypothermia was induced immediately, 0 minutes after the extinction training. One group was done after 30 minutes, and one group was done 60 minutes later. The rats were then allowed to return to their normal body temperature, and the next day they were brought into the shuttle box and tested for their fear of the black side to see if they had a memory, if they recalled that extinction process, or if they still behaved as though they hadn't experienced extinction. And they measured this by seeing how long it would take them to move from the white side of the shuttle box to the black side of the shuttle box when left to their own devices. So to orient ourselves on this graph, we have our response latency, which is how long it took them to enter that black compartment. And then across the bottom, we're going to have each of our different conditions. So the group that received no extinction training, the group that just got their fear conditioning and was left alone, they showed that they took a very, very long time here. So 600 seconds, and that's actually where they would have timed out. When they hit 600 seconds, the researchers just removed them. So they took them out of the shuttle box and they called that the end of the trial. So most rats in this no extinction procedure just never went to the black side of the shuttle box, and that's why that number is so high. For the group that received extinction but no hypothermia, it only took them 200 seconds to move from one side to the other. They went into the black box fairly quickly. So these are our 2 control groups where we can see what happens if they had no extinction and what happens if they do have extinction, and then we can look at our different hypothermia groups. So our hypo 0 are the ones that were immediately given hypothermia. Hypo 30 is the ones that had hypothermia after 30 minutes, and hypo 60 are the ones that were given hypothermia after 60 minutes. And we can see that our hypo 0 and hypo 30 both behaved like the group that had received no extinction. So this hypothermia disrupted consolidation in these two groups. But our hypo 60 behaved just like the normal extinction group. So this suggests that maybe there was some kind of consolidation failure in these groups, where experiencing hypothermia would have interfered with their ability to store what they had just learned. So maybe the hypothermia kept them from consolidating that information and putting it into long-term storage. But if you recall, at the very beginning, I had actually pointed out that there were two possible explanations for retrograde amnesia. Up here, I was looking at, is it an issue with consolidation, or is it an issue with retrieval? As I just presented it, we could say that these two groups showed some kind of disruption of the acquisition of that original memory. Being exposed to those very cold temperatures prevented them from learning or consolidating anything about the extinction procedure that they had just been through. Our alternative is, what if it's actually a failure of retrieval? What if the disruption from hypothermia was just making it difficult for new memories to be recovered? What if it's actually an issue with the retrieval phase and not actually an issue with consolidation? Well, how can we tell the difference between the two? We would have to find some kind of experimental setup, a procedure that could help us maybe reactivate a memory that has been forgotten. Now that sounds an awful lot like some of the earlier procedures that we just talked about using infants. So what if we introduced some retrieval cues? What if we made the testing procedure match the environment that they were in when they were consolidating that information? That might help us pick apart which explanation is at play here. So for our predictions, if it was a consolidation failure, if it was something wrong with our original acquisition of that information, then the information could never be recovered. Because we didn't learn it in the first place, it was never put into storage, so nothing we do should help retrieve that information. It should be lost forever. But if it's actually a failure in the retrieval side of things, then we might be able to reverse the amnesia from the hypothermia, and we might actually be able to reactivate that memory. Because the problem here is just in retrieving that information, getting to where it's stored in our long-term storage. So if we can introduce enough contextual cues, if it's a problem with retrieval, then we might actually be able to still get to that information. And so we're going to look at a follow-up to that rats with hypothermia setup, and we're going to see if we can adjust things to make it so that the rats show that they had learned that extinction. So this is our original setup. We have steps 1, 2, and 3, the extinction, and then some were given hypothermia and some were not. But we're going to see if we can go on this retrieval failure hypothesis side of things. And we're saying, okay, well, maybe the memory for that extinction process was encoded in the context of lowered body temperature. So maybe if we lower the rat's body temperature, we're going to give them another retrieval cue, and they might show that they actually did encode that information. They just didn't remember it while their body temperature was too high. So they introduced retroactive hypothermia for 3 new groups. The hypothermia treatment was given at 0 minutes because in the previous study, that group had shown amnesia for extinction. And then when they were going to test the next day, they recooled all 3 groups and tested them as they warmed up. So they were originally cooled to 30 degrees, then they tested as they warmed up to 33 degrees, and then up to 37 degrees. And 37 degrees is actually normal body temperature for a rat, so this would be when they had gotten back to being fully warmed. And so we can look at the data here. This was the original study. This is our follow-up. We still have what they did when they had no extinction and extinction. And this would be extinction with hypothermia immediately thereafter, but not tested while they were being cooled, so they showed really high again. That would be the same as this group here. And then these are our new groups. So when they were recooled and their body temperature was 30 degrees, they're still capable of moving around. They put the rats at 30 degrees into the shuttle box, and the rats showed a much lower latency to going onto the side of the cage that should have been associated with the shock. So this is showing that they did remember that extinction. And this group here, 33 degrees, a little bit warmer, they're showing still, they still went to the black side of the shuttle box. They remembered the extinction. It's just sort of a slightly higher response latency. It took them just a little bit longer. And then by the time they get back up to 37 degrees, that's pretty much the same as this group that wasn't recooled. So we're showing that, yeah, it seems that it was actually a retrieval failure. It wasn't consolidation failure, because by reintroducing those retrieval cues, by lowering their body temperature, they seem to spontaneously remember that extinction did happen. Which apparently I had written out words for as well, but I said it out loud, so it's all there. All right, so that brings us to the end of this chapter. So we can do some very brief conclusions for what we've learned about memory mechanisms. So we've said that the study of memory processes is central to the understanding of comparative cognition. What we can and can't remember and how we do and don't remember things is really important when we're studying learning and behavior. So it's a very central and core concept, and having a good understanding of what information we recall and how we store it and how we put it into storage is all very important. So our three processes, acquisition, retention, and retrieval, relate to pretty much everything when it comes to comparative cognition. Our memory processes depend on all three of those stages, and a difficulty at any one of those stages can result in failures of memory or forgetting or some other kind of retrieval problem. So hopefully having some examples, some studies to go over and chat about for each of these helped enhance your understanding of the topic and will help with your retrieval of the information on future testing situations.